Hey guys, I'm back. Second video. As you can see, we're going to talk about neuroimaging. Again, I'll try to keep it short, but I want to reassure you that, again, I'm trying to communicate just the most important information, and I'm not going into a lot of depth because it's not necessary, and I don't want to make those videos in a way intimidating. This video should be your introduction to sports neuroscience, nothing more, nothing less. So for this chapter, we're going to talk about three different techniques of neuroimaging, and then their applications to sport. What else? Yeah, let's start. So I have my notes here. So we have MRI. Okay, MRI will be the first one, and it is very essential technique when we talk about neuroimaging. And I want you to understand that the nuclear part of this relies on spin, something that's called spin. I don't want you to know a lot about that. Other than hydrogen nuclei is something that MRI is mainly focusing, and I'll make sense in a little bit. Radio frequency pulse is a field uh, that we apply when we get a bulk of the procession of all that nuclei in order to measure a signal. Okay, so we put that RF, that pulse. Now, when you apply that pulse and then you don't apply that pulse. So when you don't apply the pulse, we have something called relaxation. Okay, and there are two types of relaxation. We have the longitudinal, when the energy is transferred to the environment, the surrounding environment, and there you see that the time is called T1. So that's what we have T1 MRI, and also sometimes we have T2 MRI, and that's the transverse type of relaxation, and the energy is transferred to the nearby nuclei. So environment versus nuclei, T1 versus T2. That's what I want you to know right now about MRI, and hopefully that's not too much. Based again on spin and applying that pulse, taking that pulse away, and having two types of relaxation that we then measure T1 MRI or T2 MRI. DTI, it's an MRI, but it's a little bit more advanced, and it's based on water diffusion. And what is happening here? So we have, as you can see, isotropic diffusion and anisotropic diffusion. Isotropic diffusion is when there are no any kind of biological obstacles during the diffusion of the water, and the water diffuses equally in all directions. When there are biological obstacles and the water doesn't diffuse equally in all directions, especially in the perpendicular one, then we talk about anisotropic. And that's important when we are, in order to understand DTI, which is again a specific and advanced MRI, okay, that is based on water diffusion. fMRI, functional. So here the main goal is to visualize the brain, create images, okay, especially in those regions that have to do with information processing. As I write in my notes, it depends on bold. So bold has to do with the oxygen in the blood. So there is a comparison that happens there between oxygenated and deoxygenated blood. So the deoxygenated blood is paramagnetic. What does that mean? It's attracted when we apply an external magnetic signal. The oxygenated blood is the opposite, diamagnetic. So it doesn't, it's not attracted to that pulse. And by that difference, that's how fMRI works, and that's how we get the information that we get. Does it make sense? Deoxygenated, attracted, oxygenated blood, not attracted, when we apply that external magnetic field. All right. Now, how are these applied in sport? All right, that sounds very complicated, but how does that apply in sport? MRI. We have a lot of comparisons between athletes and non-athletes, and also athletes of different sports. We see structural alternations, such as in gray matter, right? And you know, in our brain, we talked a little bit about it in class. We have gray matter and white matter. White matter has to do more with the myelinated actions, right? And gray matter will have to do more about the cell bodies and the dendrites. But together, they create all these amazing connections in our brain, right? So we see that there is larger gray matter volumes in athletes, okay, and also higher cerebellar gray matter volume in power than endurance athletes. I cannot give you exactly what does that mean, but again, I'm telling you that the gray matter has to do with the dendrites, okay, and it has to do with more cell bodies. So we'll have more cell bodies and more dendrites. We'll have an increase of that in those specific groups. DTI. All right, let's see what's up with DTI. So here we're talking about micro structural brain changes, and we have found, for example, that specific training creates specific neuroplastic alternations in the brain, and also when there's training in old age, again we have beneficial effects. fMRI, functional changes for sure we have found thanks to athletic activity. Most of the research, as I say, is about experts, and I'm going to talk about that in a second. We know that long-term training will influence neural activation, okay? We know that AON, Activation Observation Network, will have higher activation in athletes, in expert athletes, and that has to do, AON is related to prediction, so that's important. Experts rely more on body awareness than novices, and when we talk about sports, again, shooting is very popular. The level of expertise influences neural activation, which means the higher the level, the greater the efficiency, okay? Which means lower activation. There's more efficiency. They do more with less activation. Now, what can we do in the future? Most of the studies we talk about 10, 15, 20 people. So we need larger samples so we can get more power, okay, in the results. Now, there are, as we said, techniques that have to do with the function of the brain, right? And we talked about techniques, imaging techniques, right? That have to do with structure of the brain. It would be great if in the same studies we had different types of techniques that then we can investigate the function and the structure at the same time. Applying MRI while exercising, of course, is very challenging, and it would be great if at some point we have mobile portable devices. And the last one, we have a lot of research that has to do with expertise and different levels, but we need more based on the types. So to see if there is any intersport variability. I want to close this chapter by saying that the human brain is astounding, okay? And there's so many connections, and we've talked about it in class. And this neuroimaging is one way to figure out the function again and the structure of the brain. I hope you understood a little bit the basics, because I know for some of you they are mentioned in your papers, and hopefully this helps a little bit towards your goal, which is of course to kill it with your presentation. That's all for this week. I try to keep the videos around 10 minutes, which is great, and hope you enjoy them. Let me know if you need anything, right? Again, keep lifting intentionally, not habitually. Stay healthy, stay safe. Bye guys.